Welcome to your Physiology Lab number 1. Before we get started, let me give you a little bit of an overview of how these labs will work. So what you have in front of you is the first lab out of a lab manual, a traditional lab manual that we would be using on site. Now this is an online class, and so things will be slightly different, but I still want to use the same handouts that we would normally use in the traditional lab setting. So each lab is going to have 3 parts to it. For each lab, you get a handout posted, and it has all the components that you need. And so these 3 components that are in each lab are, first of all, the important terms, and that's what you see right here in front of you. Each lab starts out with a list of important terms that I will explain to you in a video, like the one that we're just starting right now. The next part of each lab is going to be the instructions of how to start the experiments. And there will be quite a few changes to that because many things you will not be able to do just at home. But there are many things that you can do at home, and so we'll just work with this. And there will also be worksheets and things that you would have to do in activities. The last part of each lab will be the data sheet part. That's where you record your data. And then there will be also some questions. Now, that part, I will also give you then the answers to these questions, and I will go over with you over what you need to know, what you should have gotten out of each lab. And also each lab will wrap up with sort of a discussion of what to expect on the quiz that you will take on these labs. So that said, here is your lab handout number 1. And you also will need an appendix to do this lab, because for the metrics calculations, you should have this appendix handy, and I will show you that in a second. But we'll start with the important terms page, and very fittingly, our first term that you should know is physiology. Now, physio means body, and ology means study of. So in this case, we have the study of the body. That will be what this term literally means. And of course, we're here in human physiology. So we're studying the human body, and that is the function of the human body. Physiology is a science, and therefore we follow something called the scientific method. Now, that's not something a smart person came up with and then dictated on everybody that this is how you have to do science. It's more like an intuitive set of events that we naturally follow. And it always starts out with an observation. You can, even the youngest of children, you can watch them how they even follow this scientific method. You can watch a toddler on a blanket picking up, let's say, a small toy and letting it drop, and then picking it up again and dropping it on the blanket again. Basically, they're making an observation that when I drop something, it falls down, and they might wonder, does it always fall down, or does it sometimes go sideways, or maybe up, or does it do something other funny? And so they will run experiments. They will pick up this toy and drop it until they're reasonably sure that, yeah, every time I drop it, it goes down. So that's basically the scientific method in a nutshell right there. So we always need to make observations. Scientists are people that make observations, and then we wonder why something is a certain way. So I don't particularly like the order in which this is here listed, but so we're starting out with observation right here, and then we're doing a hypothesis. So that is our first guess of finding some sort of an explanation. And a hypothesis is basically you are educated, or maybe not even educated, just some wild guess of why something is a certain way. You might wonder why leaves are green, and you think, well, maybe it's because it looks pretty, or because it has something to do with the function of plants. And so you can form a detailed hypothesis or maybe a very general hypothesis, but a hypothesis is just a statement, a sort of an initial guess or explanation of why a certain observation has been made. And the only criterion that qualifies a hypothesis as a good hypothesis is that it has to be testable. In other words, we have to be able to run experiments to verify the hypothesis. So you can make the wildest of guesses. As long as it is testable by experimentation, you're good. If you form a hypothesis that is not testable by experiment, that is not a good hypothesis. And we can't work with that in science because we need to be able to measure things in science. So we're dealing with the physical world, and once you're leaving the physical world, like with some more spiritual kind of hypotheses of why something might occur, that's when we can't test it, and then it's not a good hypothesis. So in science, we stay in the physical, measurable world, and we make observations, hopefully lots of observations, and then we try to find explanations, and these initial first guesses of an explanation is called a hypothesis. And after you have a hypothesis, you're going to go to the lab and you do experimentation. So we're going to be going back up here. In an experiment, ideally you want to just change one variable to see if something has a certain effect on something else. So the experimental variable, that's the variable you want to change during the experiment and see if something happens. Here the term is also listed physiological variable. That would be, for example, your heart rate or your blood sugar levels or body temperature or something like that. It's a physiological variable. At any rate, once you run experiments, in physiology, we always have two groups. We have a control group and an experimental group. Now, in physiology, so the human body, we're not robots. We are people that are different from one another, and our bodies respond differently to certain parameters or to changes. And so if you want to test whether something has a certain effect on the human body, in this case, you can't just have one person that you're testing something out with, because that person might be responding in a certain way. It might be different from how another person would react. So you need to form groups. And the control group is the group that doesn't get exposed to a certain treatment. The experimental group, you're changing one parameter. And then you will measure whether on average, taking the whole group into consideration, whether on average there was a change. So whether there was an effect from the treatment. So did the experimental group show a significant difference from the control group? And then you can make certain statements. Now, once you're dealing with groups, you have to form averages, and you're going to have to do some statistical analysis, and that will be mostly the focus of lab number 2, which is basically sort of the lab 1 and 2, they really go together. Now, in these groups, the sample size is very important because there will be differences among the members of the groups, and in order to be reasonably sure and run meaningful statistical analyses, you need to have a good sample size. Now, of course, the bigger the sample or the group size is the better it will be for the data, but we are limited by money and by time. You can't just have infinitely large groups. So I used to do a lot of experiments at UCI with rats, and sort of a good compromise is to have groups of 10 rats that you're exposing to certain conditions and do experiments with. Or if you want to test a new medication, you probably have to have much larger sample sizes. So when they do clinical trials with people, involving people, they usually have much larger sample sizes. But at any rate, the most important thing is that you cannot be too small on your sample size, because then your statistical analysis gets very tricky. The next term is blind or double-blind experiment. Blind means that the group that's exposed to the treatment or the control group even, they don't know who is in which group. So you have a blind experiment. That means the groups, they don't know the members of the group don't know which group they belong to. Now, double-blind means that the person that's administering the test or doing the experiment doesn't know whether a person belongs to the experimental or to the control group either. So in other words, let's say you're testing a new medication, and then neither the doctor nor the patients or the participants in the study know whether they are getting a placebo or the real medication to be tested. So that leads us here to the next term. The placebo is a lookalike. It's a fake. It's something that looks like the real medication, or like a pill, a tablet or something, but there's nothing in it. It's just maybe some lactose or something like that. Next set of terms is qualitative versus quantitative observation. Qualitative means that you sort of make an observation with your senses. You see something, you hear something, you touch something, you feel it. So that's a qualitative observation. Quantitative means that you can make a quantitative statement about it. So you're measuring something by amount. Okay. Now here a little bit more detail on the hypothesis, a positive statement versus negative statement. Basically, you're saying whether something is a certain way because, or something is not a certain way because. And so you remember, hypothesis is always that you're finding some sort of initial explanation of why something is a certain way. Okay. The burden of proof in science is always on the person making a claim. If you're saying that your new medication causes people to lose weight or to grow hair back or whatever it is, and you're making a claim that you can cure cancer or lower blood pressure or whatever it is, then you need to prove that with data. So you need to run all your experiments, do all your analyses, and then you need to show everybody the data. You need to prove to the scientific community that there is really an effect, and you need to show that with numbers. Okay, so now let's move on here to the metric system, and I have quite a few worksheets for you to do on the metric system, so we'll do some separate things on this. The conversions from the English system to the metric system, there's not much you need to know, and if you were to ever be asked to convert, let's say, from inches to centimeters or something like that, and I would always give you the conversion formula, so you don't have to worry about memorizing conversion formulas. If you should need them, you will be given them, but sometimes occurs is that you have to convert from Celsius to Fahrenheit, and then you will be given the conversions formulas for that. So that said, we should kind of think about real quick, why is it necessary to do the metric system? And many of you are not super familiar with the metric system. You might find it annoying, but in science, there's no way we could do our measurements with the English system. Because think about it, in biology, things are way smaller than an inch, and if you can only cut an inch in half, and you have half an inch, quarter inch, eighth of an inch, and then you're running out of ways of describing the length of something. So the metric system is much preferred because we go in fractions or multiples of 10 on the base unit. And so you will have, in the metric system, you have the meter for length, you have the liter for volume, and you have the gram for mass. And then you go up and down with your decimal places, and I'll show you the worksheets that you need to know for that. Let's finish up here on our first recording with the microscope. For the microscope, you only need to know the names and functions of some of the most important parts of the microscope. We don't do a lot of microscopy in physiology. It really only comes up twice, to be honest. So it's the blood labs, there's some microscopy in there. And then also at first for the microscope, we have a little microscope exercise in the first lab, but since you're not on site, you will not be able to do those. And so I just want to review quickly some of the most important names of the microscope parts. The three principles of microscopy include magnification, resolution, and contrast. You must have all three in order to produce a good image. Obviously, we want to magnify something, but we also need to have the ability to resolve the image very well. So that means we need to be able to distinguish 2 points that are very close together. Contrast means that you are able to view something against a background. So if you have clear cells and a clear microscope slide, you do need to add some contrast dye in order to see something, because clear versus against clear doesn't produce any kind of image. The total magnification is always calculated by multiplying the objective lens times the ocular lens. So let's say usually the ocular lens is 10x. That's pretty typical. And then times the objective lens. Let's say you have the 40x objective lens in, then your total magnification would be 400x. Now the depth of field is when you're scrolling with your fine focus knob through the depth of your specimen. And then the field diameter is what you actually can see. Let's say you're viewing an object at 100x total magnification, and typically your field diameter is about 2 millimeters. Here's an image of a microscope that you find on page 8 of the handout, and I circled here or underlined the parts that you should know about the microscope. So please review that and make sure that you can name the parts of the microscope. So here, scrolling through this